Hello everyone, I'm happy and honored to present the work I've been doing during my PhD at the University of Geneva, which is on the modeling of fibrinolysis with the lattice-Boltzmann method using a software that we develop in the lab, which is Palabos for the computational fluid dynamics. This project is developed under the scope of the INSIST European project, which is basically a project that aims at improving the treatment of patients that have had ischemic strokes through computer modeling. So the main actor of a stroke is the thrombus. So thrombi are part of the natural healing process of the body when we have an injured blood vessel. Platelets that are present in the blood plasma bind together and activate, and they will bind also fibrinogen that will convert into fibrin and that will hold everything together, with also the addition of trapped red blood cells that are in the area. So we are happy that this happens. Otherwise, if you had a very small injury, you could have an hemorrhage almost all the time. But when this process is dysregulated, for instance, if you have too much accumulation of blood clots or clotting on the walls of the vessels, and you have, by misfortune, what we call an emboli or a thrombus that wanders in this vessel, you will have a blockage of the artery, and this will prevent thus a good irrigation of the surrounding tissue of this vessel. So this is, of course, to avoid. But when this happens in the brain, it is critical because as time goes and we do not have enough irrigation in the tissue, the cells will die, and so we can lose very quickly neurological functions and be impaired for life, or even in the worst case, die. So the two treatments that are available to us, that actually are most of the time paired together, is the injection of a lytic drug that will break down the thrombus, basically, and this is the thrombolysis. And or we can do a surgical operation. With a stent, we will pull out the thrombus from the vessel. So these are some figures for stroke, and these figures are not improving because of the aging of the population due to the improvement of our life quality. So one of the goals of the INSIST project is to improve the quality of the treatment. So to do that, we generate virtual populations of patients based on anonymous data that was collected during several years, and we simulate treatment on these patients thanks to computer models of each step of the treatment. And finally, we assess the outcome of the treatment. So in the first phase, this should be done on real anonymous patients from which we know if the treatment was successful or not. And eventually, the idea would be to be able to predict the treatment outcome for basically any type of patient in order to be able to do clinical trials that are in silico, so without having to experiment directly on the patient. So let's now dive into the subject. So fibrinolysis is a topic that is pretty well described at the biochemical level, but it is a little bit complex if you want to model every entity. Because if we do so, we will lose the sight of what are really the fundamental entities that are important for the reaction. So in our model, we just keep two entities, basically a surrogate for the plasmin and TPA and plasminogen, which we call the antifibrin, and the second entity is just the fibrin. So we consider clots that are made uniquely of fibrin. So the concentration of fibrin varies proportionally to the local concentration of fibrin and the local concentration of antifibrin, which is the surrogate for the lytic agent. So this proportionality constant, K1, gives basically the speed at which the lysis will occur. And the idea is to plug this simple equation with two entities in a complex computational fluid dynamics 3D simulation using lattice Boltzmann and especially Parabos, which is software that we developed at the University of Geneva. But for this, we need an accurate representation of our blood clot. So the approach that we chose takes its meaning from the following consideration. So the diameter and the length of a typical thrombus in an ischemic stroke patient is centimetric. So if one would want to represent naively the whole thrombus at the pore scale, where the radius of the fibers is approximately hundreds of nanometers, one should have, in the case of a 2D simulation, 10 to the 5 pixels in just one row of diameter. So if we are in 3D, this yields completely unrealistic simulations. But at the same time, we want to be not so coarse to miss out the heterogeneities of the thrombus, because thrombi are most of the time heterogeneous. So one should place itself at the scale between the pore scale and the macroscopic scale. So that is why we chose what we call this mesoscopic scale of the clot, where our voxels in our simulation are of the size of hundreds of micrometers. So then, based on the content of these voxels, we will choose what will be the properties of the flow through this voxel. So what we use is a method called the partially saturated method or partially bounce back method, which consists in the following. So imagine that here we have several voxels of the thrombus that we want to simulate that might be heterogeneous. So what we do in lattice Boltzmann usually is to compute the movement of fluid packets that go from one node of the lattice of the domain to another and then collide with each other. And with the case of the partially bounce back method, we impose that the fraction of fluid that goes through a region of clot will be a weighted sum of the fraction that goes through plus the fraction that bounces back from the opposite direction. And this fraction of bouncing back is what we call the solid fraction. So here we call this the model solid fraction because it has no physical meaning for the moment. It is just an arbitrary fraction of going through or bouncing back. So with this method, we obtain a default permeability. So this is the Darcy permeability, and it depends on basically this solid fraction. So then we need to plug the permeability relationship that is specific to blood clots and not just an arbitrary relationship. So what we do is basically just invert this relationship for gamma, the model solid fraction, and we need to find a mapping of gamma based on the real solid fraction of the clot. And then we can obtain the desired relationship of the permeability based on the solid fraction. So we can do this scaling with several permeability models. So we have here 3 permeability models that are used for porous media, and their implementation with this methodology in the simulation. So we see 3 cases: Davis equation, Jackson and James equation, and Clark equation, that all try to characterize the dependency of the permeability on the solid fraction of the thrombus. And in pink crosses here we have the results of experiments of permeability done by Wolfsus that measure the permeability of in vitro homogeneous blood clots. So this is Davis equation, and from the equation you see that it depends on the radius of the fibers and the solid fraction, of course. But as the lysis occurs, the radius of the fibers will become thinner and thinner, and so the permeability should be adapted with this shrinkage of the radius also. And last but not least, also the solid fraction needs to be rescaled based on the shrinkage of the fibers. So this was it for the theoretical part of the model. But then what we did actually to calibrate the model is to compare it with in vitro fibrinolysis experiments done by our partners at the University of Brussels. Why? Because it is very hard to get in vivo data for modeling. First of all, because the patients arriving at the hospital need to be treated as fast as possible, and there is no time to take measurements that are unnecessary for the emergency treatment. Also because it is unlikely to have at ready the measurement apparatus, for example, to measure the blood pressure before and after the thrombus in the patient, or to measure the permeabilities of the clots, or even to characterize precisely the composition of the thrombus. Also, the patient is already in a stress and shock situation, and it might be wise to consider if it's really necessary and ethical to do more experimentation on a patient that is already, let's say, almost dying. Also, if one wants to build a model, it's important to have consistent data that can be reproduced to test back the model. And since these ischemic stroke events are generated naturally within the patient, it's impossible to replicate the same event and to try to predict it. There are other points that prevent us to get clinical data that is relevant for modeling, but I think these points are already sufficient to understand. So that is why we went to controlled in vitro fibrinolysis experiments. Homogeneous fibrin clots were created in those tubes, and we poured a solution that contains TPA above of the clot, and we just have this gravity-driven flow that makes this solution perfuse slowly through the clot. As the solution gets perfused, we add again a level of solution such that we keep the level constant. So this picture is just before adding back more solution. So experiments were made, a variation of the initial concentration of fibrin, TPA, and initial pressure drop. Basically, we just added more solution to get a higher hydrostatic pressure. So we then replicate this in the model. We apply the same pressure drop between both ends of the tube. We put our homogeneous clot that is made of these partially bounce back voxels, and we put a solution that will contain our anti-fibrin particles that represent the TPA and everything that works to lyse the fibrin clot. So to validate our model, we considered the following measure. We saw that in the case of homogeneous fibrin clots, there was a clear distinction between the solution and the clot. So we could measure accurately enough the position of the front of lysis. So after calibration of our model, we obtained the following results for a variation of fibrin in the top row. So here we have experiments with initial concentration of fibrin of 2 milligram per milliliter, here 3 and here 4, and we see that with the same reaction constant that has been calibrated, we get pretty good agreement with the simulation data, which is in blue, compared to the experiments that are in orange and green. Here, with the variation of TPA, we also get very good agreement, and also with the difference of pressure. So, given the initial conditions of the experiments, we were able to predict the lysis front position in time. So, of course, there are several limitations to this model. So for now it just comprehends homogeneous clots, but it is possible to extend it to heterogeneous clots by changing the permeability law, for example, of voxels that are richer in platelets or red blood cells, for instance. Also, we do not take into account the fragmentation of the clots, and also another limitation is that from the in vitro experiments we have rigid walls, and this might impact the apparent permeability of the clot with respect to arterial walls that are compliant and that have also pulsatile flow. So thank you for your attention. I will be happy to answer the questions that you might have.